Welcome back, everybody, to the second session of our four-part webinar series. We, as I said last week, are really very overwhelmed and excited by the incredible response that we've received for this symposium. You'll probably see on your screen right now that there is a questionnaire. If you didn't complete the questionnaire on the first lecture, we would appreciate it if you could complete the questionnaire today. And it will be available on the beginning of each lecture for people that are coming for the first time. This will just help us to try and measure knowledge and impact as we move forward through this process. So thank you very much for doing that. We really appreciate it. Also, to help facilitate communication and collaboration, we've created a WhatsApp group for all participants. You don't have to join, but if you want to, we encourage you to join to stay connected as we work together to improve snakebite treatment in East Africa. If you go into the chat, simply click on the link at the top of the chat. We'll try and post it through the evening again in case it gets lost. And that will enable you to join that East African Symposium follow-up WhatsApp group. Please, if you're posting questions today, could you try and post your questions in the Q&A box, not in the chat? It's quite hard for us to keep track of both the chat and the Q&A box. So we'll be monitoring the Q&A box if you could post your questions in there. Tonight's webinar is going to focus entirely on antivenom, a crucial yet often misunderstood aspect of snakebite management. Given the complexity and importance of this topic, we hope tonight's presentations will provide valuable insights and clarify many questions. It is my absolute pleasure to introduce our first speaker, Dr. David Williams, who will share his extensive experience and provide a comprehensive overview of antivenom. Dr. David Williams is a technical officer of antivenoms in the Regulation and Pre-Qualification Department at the World Health Organization. He previously served as the head of the Australian Venom Research Unit at the University of Melbourne and as the head of the Charles Campbell Toxinology Centre at the University of Papua New Guinea. Dr. Williams has over 20 years of field experience working on snakebite issues in Papua New Guinea. In 2008, he founded the Global Snakebite Initiative, advocating for snakebite envenoming to be recognized as a WHO neglected tropical disease. His work has helped raise awareness about the urgent need for sustainable solutions to save lives and prevent disability worldwide. Thank you very much, Dr. Williams, and we're excited to invite you to the stage.
Thanks very much, Ali. Good evening, everybody. I'm going to get straight into this and answer the question, what is antivenom? That's the very first thing we need to start with. And basically, they're very specialized biological medicines. They're produced by typically collecting snake venom, processing it, quantifying it, and then preparing it as an immunizing mixture that's injected into a large animal, such as a horse or a sheep or a camel, which produces antibodies to those proteins in the venom. Those proteins can then be harvested by collecting blood and plasma from the animal. Goes through quite a complicated processing process by manufacturers and might be eventually presented either as a liquid product in vials or ampules or as a lyophilized, or some would call it a freeze-dried product, both of which, again, have to go through a process of quality control, potency testing, batch characterization before they're finally released. Now, the basic pharmaceutical ingredient are these purified animal plasma-derived antibodies, but there are other substances that can be present, including stabilizing agents or preservatives, sodium chloride in the carrier fluid, and in some cases there can be unintended contaminants. Regulation, unfortunately, for these products varies quite a lot, and that can have a very direct impact on the quality, safety, and efficacy of the products. If you're in a setting where regulation is strong, then it's very likely that you'll have very safe and very effective products. In environments where regulation is not particularly mature, it can be quite the opposite. And a major thing that WHO is involved in is trying to improve the process of regulation and control of antivenoms and the surveillance of their production and use. So the next question, or the next issue to address really, I guess, is that not all antivenoms are the same. Often you get a product, it's in a 10 ml vial, it might be made by manufacturer A or manufacturer B, and many people would think that that's an identical product regardless of which manufacturer makes it or which batch of one particular manufacturer's product you happen to be looking at on the day. The truth is it can be very, very different indeed. And I'm going to sort of avoid the wording of these slides a little bit. If you focus on the diagrams, they'll probably tell you everything you need to know. The key takeaway is that you can have 6 different products and they can all contain completely different amounts of the actual active pharmaceutical ingredient, the purified antibodies, and also different amounts of other things as well. And you can see here, these are actual test results from 6 different products that in some cases they have less than 1% total protein in them. In others, the third one along, you'll see it's around about 10.2 or 10 to 12%, but it can be quite variable. And some of them contain very little actual active pharmaceutical ingredient and a lot of other stuff, which indicates that those products may not be particularly useful for actually treating snake bites. Now, most manufacturers claim that they will have at least 85% API among the total protein. The reality is most of those products don't actually reach that standard. And again, this is an issue related to the poor historical regulation of these products. So moving on, again, to the same topic, but with a little bit more detail. It's not just a matter of how much product is in the vial. It's also a matter of, well, what are the products that are in the vial? Are they the actual pharmaceutical ingredient? And in this case, I've shown those same 6 products. 4 of them are what we call FAB prime 2, which means that the FC region of the antibody, about a third of the antibody in size, has been chemically cleaved away and discarded, whereas 2 of the others are actual intact IgG that have the full size molecule. The active pharmaceutical ingredient is shown in dark green in each. You can see that the FAB prime 2 products, none of them actually reach that 85% mark, whereas the 2 whole IgGs, one of them actually exceeds it, one of them gets very close to it. You'll also notice that the contaminants, the other things that are in the product, is much more reduced in the whole IgG compared to the proportions of, for example, small molecular weight fragments shown in orange, or non-immunoglobulin proteins such as antithrombin 3, alpha-2 macroglobulins, fibrinogen, alpha-1b glycoproteins, and other things that could potentially be responsible for the adverse reactions that some people have to antivenom. And of course, again, strengthening regulation and control will eventually improve the quality of these products and get them to meet the specifications that they need to, so that they can be both safe and effective. Now, at the same time, not all snake venoms are the same either. And if there's any takeaway message from this, it's that I want you to understand that there's not a one-size-fits-all approach when it comes to dealing with snake bites and antivenom treatment. Each of the different types of snakes can produce different amounts of venom, and I've grouped 4 major groups here: the carpet vipers, the puff adders, mambas, and the spitting cobras, and shown you the relative amounts of venom that they each produce. And interestingly enough, it's not just the volume, but it's also the concentration. So for example, puff adder venom has about 17% more molecules per milligram than carpet viper venom does. But on the other hand, black mamba venom has 196 times more molecules than an equivalent amount of small-scale viper venom would. So the more molecules there are, the more antibodies are needed to neutralize them, because an antibody has 2 functional binding sites if it's FAB prime 2 or if it's whole IgG. So for every molecule, you're only going to bind 2 molecules of toxin. The more molecules of toxin there are, follows that the more molecules of antibody and the more vials of antivenom you may actually need. Now, if we have a good antivenom, a product that, say, has around about 350 milligrams of functional antibodies in it, those are not all antibodies that are going to actually neutralize snake venom. It varies between around about 5 and 20% depending on the species of snake. So most antivenoms have an average of about 5.7% neutralizing antibodies against carpet vipers. And in that case, a product with a total of 350 milligrams, you might only need between 1 and 1.6 vials, so 1 or 2 vials. On the other hand, for a black-necked spitting cobra, where there's about 11.2% neutralizing antibodies, you could need between 5 and 20 vials. And for a mamba, anywhere from 3.5 to 5.5. Now that's, again, a good product with quite a large amount of antibody in it. But what happens if you get a product that doesn't have much total antibody in it? And so, for example, if we had a product that only had 75 milligrams, and again, assuming in an ideal world the same percentages of neutralizing antibodies, instead of 1 to 2 vials for your carpet viper, you're going to need between 3.5 and 7.5. And for that black-necked spitting cobra, you're going to need somewhere between 26 and 94. In other words, this antivenom is going to be a clinical failure because you physically cannot administer such a large number of vials to a person. You know, at 93 vials, you're talking about almost a litre of antivenom. So it's straight away that product is a failure based on that alone. Now, I'm only going to give one sort of slide around the actual administration of antivenom. And I know for health workers, it's something that they ask about a lot. So antivenoms need to be administered as soon as possible once you've been able to observe signs or symptoms of envenoming. They should be given either one way or the other. So intravenous infusion, using a bag of intravenous fluid as the carrier, or you can use the intravenous push method using a suitable sized needle and syringe. Now, when it comes to what you're going to do about giving it, one of the issues that often comes up is the question of pre-medication. And the use of subcutaneous adrenaline in very small doses, about 0.25 of a milligram, subcutaneous about 5 or 10 minutes before you start antivenom, varies from one place to another. I think if I remember rightly, South African guidelines don't recommend its use at all. In Papua New Guinea, we used it extensively. And in my experience, it did reduce the rate of early adverse reaction, and it was safe to give to patients, whether they were small children, the elderly, pregnant mothers, etc. And like I said, we saw around about a 75% reduction in adverse reactions against what had been happening historically when we used it. It's also important that you have additional adrenaline drawn up for use to treat an early adverse reaction. It's the #1 drug for dealing with early adverse reactions. But rather than subcutaneously, if the person's having a reaction, you would give it via the intramuscular route, which is going to be far more effective, more quickly for dealing with that reaction. Things like hydrocortisone don't have any role. Antihistamines can be potentially useful, but some of them can be sedative. So it's probably best to actually titrate them to ease itchy rashes and problems that are causing discomfort to people in that sense. All these slides will be available from the organisers afterwards. So like I said, I won't go into too much detail of reading every last thing, but the schematics give you a little bit of an idea of how the product can be prepared. So if you're going to do it by IV infusion, you might take 5 to 10 vials, so 50 to 100 millilitres, dilute that to a total volume of 200 mils in a small intravenous bag or a paediatric burette. And after pre-medication, it can be infused at a rate of about 30 minutes. And so if you're using a standard adult giving set, you're going to need around about 34 drops every 15 seconds to be able to run that volume through in 30 minutes. If you're using a paediatric giving set, that'll be around about 120 drops, I think, a minute, every 15 seconds, 400 a minute. If you're using IV push, and IV push does have some good advantages. One thing, it keeps the doctor at the bedside right throughout the administration. And so again, you might take a couple of 50 ml syringes, draw up 5 to 10 vials worth of antivenom, undiluted, and then it's slowly given at a rate of 2 mL per minute by the medical officer intravenously. So it's an intravenous push injection. The benefit of that is if the person starts to have a reaction, it's very easy to just stop pushing, treat the reaction, and then once it settles down again, you're able to continue. But that's all I'll sort of cover with that today, because I know Professor Worrell is going to deal with this more extensively when he speaks in a week or so. So now let's go back to snake venoms. And a couple of things that you really need to understand is that even in the same species of snake, the venoms can be vastly different in their composition and in their potency. And so here I'm going to give you an example of an Egyptian cobra on the left-hand side from Gulu in northern Uganda. And on the right-hand side, we've got an Egyptian cobra from Athi River near Nairobi. Now, the snakes from Gulu, this is what we call a high-pressure chromatograph tracing. It shows the individual components. These big peaks to the left at the Gulu snakes, they're all neurotoxins. But then when you come and you look where the Athi River snakes are, you'll see that on the left-hand side of that trace, there are virtually no neurotoxins at all. And whereas you only need 1.84 micrograms of venom per mouse for the LD50 in Gulu, you need 65.7 micrograms a mouse for the same species of snake if it comes from Athi River. So very much less potent, almost no neurotoxic effects at all, whereas the snakes from Uganda are extremely potent. And even in some mixed populations, so if you're in Kenya and you go to Naivasha, you'll find Egyptian cobras there. Some of them don't have neurotoxins, some of them do. And when they have babies, Mendelian genetics is what ensues. You end up with a mixture of snakes with all neurotoxin, no neurotoxin, and something in between from the same two parent animals. So composition, potency are important, but so too is venom yield. And one thing that doesn't often get considered by the manufacturers of antivenoms is actually how much venom a snake may inject that may need to be neutralized by antivenom. And again, given that different snakes produce different venoms with different molecular sizes, different numbers of molecules, the dose of antivenom is going to vary according to how much they can inject. And so in defensive bites, we found that on average, carpet vipers are about 6.5 to 14.5 milligrams. For puff adders, it's about 60.3 to 108. Black mambas, 37 to 58.6. And for Nigrocollis, the blackneck spitting cobra, about 43.8 to 158.9, which is a lot less for a single defensive bite than what we can get when we physically restrain them, as I'm doing with this, I think it's a forest cobra in the picture with Bonnie from the snake farm. We can actually collect much larger volumes. You can see that when we do that with a large cobra, we can get almost 500 milligrams. From a puff adder, we can get about 150. And from mambas, we can get up to about the maximum 338.2. So quite large amounts. And as well as the volume of venom being different, the potency and the composition being different, the biting behavior is also different. So I'm going to show you two very quick videos. The first one is a black mamba, and these are slowed down to about 60 frames a second so that you can actually see the mechanism that goes into a bite. So mambas tend to bite very quickly. They may deliver single bites in rapid succession, maybe 2 or 3 times. And so this is what that looks like, and I hope you're all able to see it properly. So mouth open, and then it's going to be a quick stabbing bite, and he's going to release it so that whatever he's bitten can't potentially harm him. If we then have a look at another snake, and on the right-hand side, this is Najahaji, an Egyptian cobra. Their biting behavior is quite different. This snake is going to grab hold of the container, and then it's going to bite down very hard and chew. And in a minute, you'll actually see venom running down the side of the container. So this is potentially why they can deliver much larger volumes depending on their biting behavior. So if you have a patient, for example, who says, the snake gave me one very quick bite and then let me go and run away, may not necessarily have had time to inject any or very much venom. On the other hand, if somebody's trying to kill a snake with a stick and it bites them and it hangs on like a dog, chances are it's going to inject quite a large amount of venom. And these are, again, taking the history of a patient, important things to consider. Now, how do we work out what's going on with snakes? How do we work out the potential use of an antivenom? So in the WHO lab, which is based at the Institute of Biomedicine in Valencia in Spain, we will take antivenom and we will, looking from left to right, immobilize it in a particular chromatography column so that the antivenom is bound. We'll then incubate it with venom, and after a period of time, we will flush out everything that the antibodies weren't attracted to. And then what we can do is we can see that. So we can see this is the natural profile at the top middle of the venom. We can see the components that were actually captured in the column here. And we can see at the third row, some components that went straight through. We can then, for each of the individual toxins, work out what percentage of each of those toxins was bound. And some of them, it's only 35 to 40% of what we added to the column. In other cases, it might have been 100%, so very good binding. But that's using a measured amount. If we then translate that to how much of each of those toxins are produced in the venom yield of the snake proportionally, we can look down and we can see at the bottom right that the first two peaks together make up nearly 60% of the venom of this particular snake, the black mamba. But the antibodies are only binding about 10% of one of those, even though it makes up nearly 27% of the venom, and about 13% of the other, but it makes up 41%. So a lot of that venom is not going to be actually immunorecognized by a standard dose of antivenom. And again, we can calculate exactly how much venom is bound per vial, and using that we can extrapolate in theory how many vials might be needed to achieve enough of a circulating antibody volume for that antivenom to be potentially effective. And again, this gives us a lot of valuable information. Here we can see that the percentages of toxin binding antibodies for different snakes, for puff adders in green, for carpet vipers in orange, black mambas in purple, and in the pinkish mauve, the Naja nigricollis, the spitting cobra, we can see that an antivenom that has 163 milligrams of functional antibodies is going to bind quite well. But then volume of antibodies is not necessarily good, because we have another product here that's 884 milligrams, but it's actually worse at binding those particular venoms than the one that only had 163. So from that, we can take a look and we can see, well, how effective might they be in terms of the percentage of toxins bound compared to the toxins in the average venom yield. And we can see that for carpet vipers in orange, all of these products have an excess. They have maybe 100 or 200 times more antibodies than are needed. But when you look at the big blackneck spitting cobras, none of them have 100%. In fact, they barely have 10% of enough. So you're going to need a lot of vials in order to get to the point where you could potentially bind 100% of the toxins that may have been injected. And again, these are important things to take into account when we're thinking about dosing. We can also look at, well, what are the antibodies actually working against? And there's a widely stated, I guess you'd call it a factoid in the literature, that says that most antivenoms are very poorly effective against elapid venoms because elapid venoms have weak immunogenicity. It's actually not true, and the data proves it. You can see here that 3 different types of toxins in blackneck spitting cobra venom. Some of them are 10% toxin binding antibodies, quite high. Black mambas, the best one, was about 14, 15%. But then when we come and we look at the puff adders and the carpet vipers, they're only around 2 to 5% at best. So there are far more antibodies against those cobras and mambas that are available in polyvalent antivenom than there are anti-viper antibodies. But the problem is, elapid toxins are very small. They're about 6 to 9 kilodaltons in size, whereas a puff adder's venom, the average size of a molecule might be between 23 and 48 kilodaltons. So you might actually have 6 times more molecules of venom in the same quantity of mamba venom as you do in puff adder venom. And that means you're going to need 6 times the quantity of antibodies if you're going to hope that that product's going to be effective. And this is why when you see a product that says you use the same dose regardless of the species, they are telling you that based on absolutely no clinical data whatsoever. That's just a made-up number. If a product has been robustly tested, they will recommend different doses of antivenom for different types of snakes based on the characteristics of venom composition, venom yield, the potency of the venom, the size of the molecules that are in the venom. And so while it may seem to people who've been used to just using a single 2 or 3 vial dose that, oh, this product says we need 10 for this type of snake, maybe it's not as good. In actual fact, it's probably giving you far more useful information about what's needed to get clinically effective treatment than the other products that recommend far less. And all of this data we can then use to compare products. So going back to our Egyptian cobra from Uganda, we can see that one product, even though it's 163 milligrams of antibody per vial, it only has a potency of 0.272 milligrams a vial. And some of the key toxins, the key neurotoxins, the first 4 here in red, and there's 2 more at positions 8 and 17, are very poorly neutralized. All of them are 3-finger toxins, so they're either neurotoxins or cytotoxins. And the proportions that they can neutralize of the average venom yield is actually quite low for each of those toxins. So this is going to be a very poorly effective product given the low potency and a very large number of vials that you would have to use. On the other hand, we've got another product which is more antibodies, 486 milligrams per vial, has a potency 6 times higher almost, 1.2 milligrams per vial. But it's actually much worse off in terms of which toxins it actually successfully binds to. And the reality is that you may still need to use a very large number of vials and may get quite a different response. But this at least allows us to compare one product to another and make functional decisions about which one may be theoretically the best one to take forward to, say, clinical studies, clinical trials. And again, it varies. One more example. So carpet vipers. On the left, we have Echis romani, the carpet viper from Cameroon. In the middle, we can see Echis ocellatus, the carpet viper from Ghana. Now, can you guess which species the manufacturers are using to raise their antivenoms against? It's fairly obvious, given the good response of 5 different products to the Cameroonian Echis romani, that Echis romani is probably the species that they've been immunising their horses with. That very closely related and not very geographically distant Echis ocellatus against several of the major types of toxins like PLA2s, what we call phospholipases A2, C-type lectins and metalloproteinases, actually very poor immunorecognition. In some cases, less than 1%, other cases 2 to 3% at best. So quite different. And of course, these snakes have different venom yields and different potencies. Cameroonian snakes yield about 13 milligrams, whereas from Ghana it's about 11. But the Ghanaian venom, even though it's less, is more potent than that of the snakes from Cameroon. And that means when you read an antivenom manufacturer's package and it says 1 milliliter will neutralize 25 LD50s, for each of these snakes, the 25 LD50s is going to be a different volume of venom. The case of the snakes from Cameroon, it's going to be about 870 micrograms per mil. In the case of the Ghanaian snake, it's only 660. So even though the Cameroonian venom is less potent, you're going to need to use about twice the number of vials as you would for the more potent venom of the Ghanaian snake, just simply because of the combination of volume, venom composition, and potency. So again, I'm just keep going back to this message I'm trying to give you, that there is no one size fits all approach when it comes to treating snake bites. We can also use this to look at estimating theoretical doses if we're thinking about doing clinical studies. And for all of these different species of snakes and these 5 antivenoms, you can see that the first one, very low amounts of actual antibody, would be an abject failure. You'd need extraordinarily large numbers of vials to be effective against any of these snakes. Most of the others work very well for the carpet vipers and the puff adders, but they failed against the cobra venoms, and two of them were reasonably good for a couple of the populations of mambas. But you can see even 3 different populations of black mambas, the response is quite different from one antivenom to the other, and even for the same antivenom used for treating bites from different geographical areas. And that's at 100% of the average venom yield. Looking at 50%, the results are still bad. And when we do this testing in vitro, so we're doing this in I guess you call it a test tube environment, this is overestimating what's going to happen when we start testing in live animals. And so if we go and very quickly look at what happens with live animals, you'll see that the results against the cobras and mambas is suddenly actually much worse when you're testing a real biological system at either 50 or 100%. So we know that what we can learn from in vitro studies is going to be an overestimate, and that what's going to happen in practice is going to be somewhat worse than that. But it does at least give us a starting point. It tells us, well, at a minimum, we need to start with this sort of dose and be prepared to work upwards until we find the right dose. So that can be useful for trial design and for working out where you're going to start. And again, I'll go back again to the concentration of functional antibodies and vial estimates. And so we can see here that product that was 163 milligrams. For these 4 species, you're going to need, for a puff adder, somewhere about 15 vials. For a carpet viper, you're going to need 3 vials. For a black mamba, it's probably going to be somewhere between 7 and 8. And for Naja nigricollis, it could be more than 30 if you look at the error bars. For a product that has a lot more antibodies, you're going to need much fewer vials because there are more functional antibodies available. In this case, this product would potentially be useful at less than 10 vials for each. The product that's in the middle is going to be literally in the middle. It's going to give you somewhere between 1 to 5 vials for your puff adders, your carpet vipers, and your mambas. But when it comes to the cobra venoms, which are much more voluminous, you're still going to be talking at somewhere between 10 and 20 vials, perhaps. So moving on to the next, I'll just shift to, well, what is WHO doing about all of this? And we developed a strategy in 2019, a roadmap, if you want to call it that. This roadmap has 4 key pillars, one around engaging and empowering communities, another one around ensuring safe, effective treatments, strengthening health systems, and then partnerships, coordination, and resources. So we've been working on making safe, effective antivenoms available and accessible, improving control and regulation, and pre-qualification. And so a lot of my work is around what's called a risk-benefit assessment of current antivenom products, where we review information received from manufacturers. We do independent laboratory assessments. We inspect them to see if they're GMP compliant, that's good manufacturing practices. And then eventually a committee will make a decision on whether the product should be recommended for use or not. It goes through a formal application process. There's information on our website about how that process works. They have to provide us with samples of the venoms they use, as well as the antivenoms themselves, so they can go through robust testing. we don't just test their snake venoms, we test snake venoms from probably, for each species, maybe 4 or 5 locations. At present, we have about 10 assessments in progress in sub-Saharan Africa. We've got 9 in progress in the Middle East and North Africa, and we've got 8 in progress in what we call South Asia, which includes Pakistan as well as right through to India, Nepal, Bhutan and Sri Lanka. So quite a lot. Now, this is a very extensive process. The dossier review is about 30 to 60 days. Depending on the complexity of the product, if it's the number of venoms it's meant to neutralise, it could take 6 to 9 months or more to do the laboratory evaluation. GMP inspections, we may do the initial one in a week, but then addressing the issues that we uncover could take a manufacturer between 12 and 18 months, and in some cases substantially longer. Then goes to the committee. They will need time to consider all of the data, so it's about a 30-day process. And then there's another process around notification. We don't just come out and say, well, here's the result. We will first of all write to the manufacturer and share what the recommendation is likely to be, and we give them a right of reply to defend the position they want to take. And then, of course, our senior management will look at that. They will make a final decision, and by about 90 days after the whole process has been through, we'll be able to notify the public of what the results are. So depending on the process, it could take anywhere from 2 to 3 years. We have some products that we initially did GMP assessment of in 2017. We're 8 years down the track and those manufacturers are still no closer to fixing their issues. So it can be a lot more laborious, unfortunately. In the laboratory, we're very comprehensive. We look at the physicochemical properties of the antivenom against manufacturer specifications. We look at the composition of the product with a range of techniques to determine exactly what may be in it that shouldn't be there and how much of the right API is present. We then do the in vitro work, immunorecognition analysis, what we call 3G antivenomics, and only if we get good results with that will we then proceed to test in animals. And we test not just the overall potency, but we test various species-specific activities such as hemorrhagic activity, procoagulant activity, myotoxic activity, and so on. And again, all of this information helps to inform the final decision we reach. Lots of products from lots of parts of the world. What are we trying to establish? Well, there's really 5 things. We're trying to determine that the product as it's designed and formulated is going to be safe and effective where it's marketed. The quality of the product as it's produced, it's going to be quality assured according to international standards of GMP. We want to know, is there independent laboratory evidence going to support the manufacturer's claim about the laboratory? So that's where our lab comes in. And then we want to know, well, what is the available clinical experience? And in many cases, these products have never been through clinical trials. But what we're trying to determine is, well, on balance, is there enough information to show that these products are being effective and safe, or to the contrary, does the real world evidence show something else? And of course, we're also interested in integrity. Has the manufacturer been open, honest, and transparent with us? And are they cooperating with us? And unfortunately, in the past, we've had instances where that's not necessarily been the case, and that can slow the process down. Our technical advisory group includes veterinarians, experts in GMP production, biochemistry of snake venoms and preclinical quality assessment, clinical medicine experts, people with expertise in biological standardization of these types of products, clinical and quality assessment of biologicals, production and purification of antibodies, things like viral validation, and design and conduct of clinical trials. And we have, I think, 8 members at the moment, and we're going to expand that to about 15 this year. So we will be announcing a new call for additional nominations from national regulatory authorities, national control laboratories, and academic institutions in 2025. Finally, just to get to the end, 3 product assessments for Africa have been completed and products recommended. The monovalent microfarm product Equitab G for treating carpet viper bites in West Africa. Anti-Vitamin Africa for treating carpet viper and puff adder bites. It's not currently recommended for mambas and cobras, but we have new data from the manufacturer that's being assessed at the moment. It may well be possible at certain doses that it would be effective for those snakes. So that recommendation may be improved upon soon. And then the one that is widely recommended at the moment is PanAF Premium from Premium Serums and Vaccines. We have 10 more that we expect to finish this year. Equitab Plus ICP from ICP in Costa Rica, 3 different products from Biological E in India, the SAIMR product from South African venom producers, several products from VINS, their African polyvalent, their monovalent Echis, their new boom slang antivenom, and their South African polyvalent antivenom that's similar in design to SAVP's product, and also Inasan's InoCert Pan-Africa product. And so all of those assessments should be completed by the end of this year. When we're finished, they'll all be published on the website, and you'll be able to find all of the details, including public reports on the laboratory assessment, public reports on GMP inspections. So it'll be very transparently covered. These are the 3 currently recommended products. There is information on the website at this link about those products, what they're recommended for, what they're not recommended for. And you'll be able to download PDFs that contain all of the information. In some cases, in working with the manufacturers, we've had to ask them to improve their package inserts. So we want people to know how much venom you're talking about when you refer to 25 LD50s. And so you can see the amounts that equals for each species are now in the package insert. There's also supplementary information for cobra and mambas about how to deal with envenoming where antivenom may be less effective, particularly through good airway and breathing management. And in the case of cobras, potentially the use of anticholinesterases. And so I will leave it there. Like I said, there's a link to this that'll be in the PDF. You can go and visit that and learn more. But right now, I'll wrap it all up and hand it back to Ali.
Dr. Williams, thank you. That was incredible. I don't know about you guys, but I have just learned so much. I'm sure, like me, you'll want to go back and listen to all of that again when the recording becomes available next week. Yes, please stop asking. Everything is being recorded and you will have access to the recordings. We will post in the WhatsApp group. I am just putting a link in the chat. That's how you join the WhatsApp group. I will make sure that I let you know how to get those recordings, and Daktari Online are also going to tell you at the end of this evening's talk how to get those recordings. Can I ask you a few questions, Dr. Williams? I'm just picking out a few that we haven't yet answered. You did state that in South Africa, they tend to automatically, or in South Africa, they don't give adrenaline straight away, but here we do. Somebody is saying, why not treat the anaphylactic reaction when it occurs, which is what you said. So in some places they do and they don't. I guess it's just a procedural thing.
Well, look, if you've got a chance that you might be able to prevent a reaction in a poorly resourced health facility where the staff may not have the ability to actually manage severe anaphylaxis successfully, what have you got to lose?
Yeah.
If they can't manage it, the patient could die. Pure and simple.
Yeah. So the other thing was that antihistamine can cause cutaneous. Why does hydrocortisone have no role as it is an antihistamine?
Hydrocortisone is not going to do anything for probably 6 or 9 hours. Okay. You know, it's a steroid, and steroids don't treat, you know, these sorts of cutaneous eruptions. But an antihistamine, you can titrate a very small dose of it, and within 10 or 15 minutes, that person has relief from the itching that may potentially cause them to scratch out their IV line.
The concentration of adrenaline, which is co-administered with the antivenom, is it 1 to 1,000 or 1 to 10,000?
Okay, so it's an interesting question. When we first started its use in Papua New Guinea, the Paediatric Society wanted us to use 1 in 10,000 for children, and we were using 1 in 1,000 for adults at a dose of 0.25 milligrams for adults and a titrated amount for kids. But we found in actual experience that 1 in 1,000 was tolerated very well by all age groups. We looked at a lot of data on heart rate, on blood pressure, on respiration rates, body temperature, and we found that it made no difference if it was given by the subcutaneous route. And so eventually what we did is the Paediatric Society said, well, just use the same dose for children as you use for adults, but it absolutely must be given subcutaneously if you're pre-medicating. If it's an early adverse reaction, it's different. You use it intramuscularly. Different half-lives involved in it.
David, could I interrupt there and say,
yeah, sure.
You're surely not suggesting co-administration. Ali said co-administered with the antivenom. No. It's something given before, as David said.
It's 5 or 10 minutes beforehand.
Before you start antivenom. It's given by a different route. So this is a very important point, actually. It's not co-administered with the intravenous antivenom.
Yeah, and the reason for that is the half-life of subcutaneous adrenaline is around about 43 minutes. So if you're going to run your antivenom over 30 minutes, you give it 5 or 10 minutes beforehand, you're not going to reach the end of that half-life while the antivenom is still running. If you're dealing with intramuscular adrenaline, the half-life would only be 12 minutes. So,
thank you.
You know, very different.
Okay, we've three more questions. How long does the antivenom last in the body?
So it depends a little bit on where it gets distributed to, which tissue bed it goes to, and also the size of the molecules. We know with antivenom, for example, small antivenom molecules like they use in America, FAB, which is about 50 kilodaltons in size, gets flushed out by the kidneys very quickly if it's in the circulation. Whole IgG or FAB prime 2 last anywhere from 10 to 15 days because they're 2 or 3 times bigger in size. The same applies with different types of toxins. Once they're not bound and they're in the circulation, the smaller they are, the quicker they'll be eliminated. But of course, if they can get into, say, well, neurotoxins in some case that can get inside a nerve cell, they can safely sequester themselves for several days before they're eliminated.
Okay, thank you. I think I don't think we have it here, but I do think it is available, an actual test in certain countries where they can do a test to know which snake has bitten you or what venom you've been injected with.
Yes, in Australia, we have a venom detection kit that can identify each of the 5 different immunotypes from different groups of snakes, but it doesn't tell you how much venom is present. Now, in our research laboratory, we developed a similar assay that could actually tell us how much venom was present per milliliter of patient plasma. But that's more of a research tool than it is an actual clinical tool. And the problem with these is that in poor settings, the cost of such tests is exorbitant. And it's just another impost on patients. And if the product you have is a polyvalent product and it's going to be used for everything, other than for research or epidemiological purposes, there's not much point in actually really, trying to impose that cost on all these poor patients.
Yeah. And last one, and I'm pretty sure the answer is you give it anyway, but for people with sort of other conditions, cardiac, chronic conditions, renal conditions, do you give antivenom anyway? Because that's the only way to neutralize the snake venom.
Well, it is. I mean, it's the same question that comes up sometimes about pregnant women. Do we give antivenom to pregnant women? Look, you're either going to have one person die or 2 people die. You treat the mother to save them both. And the same goes if you have other comorbidities. It's important that the doctor or the health staff know about them and can manage things appropriately, but still not going to rule out necessarily, giving antivenom to actually treat them.
Noted. Thank you very, very much indeed. I think we'll move on now. Next, I'm very pleased to introduce Dr. Kadilka, the Technical Director of Premium Serums and Vaccines PBT Limited, who produce various therapeutic equine immunoglobulin products, including the WHO approved premium Panaf antivenom that we're currently using here in East Africa. Tonight, Dr. Kadilka will give us a better understanding of this product. Dr. Kadilka earned his degree in veterinary sciences in 1985 and completed an MSc in veterinary medicine from Bombay Veterinary College in 1988. He has also studied business management and human resources management with over 22 years of experience in antivenom, antitoxin manufacturing. His expertise spans animal healthcare management, venom production, quality management, applied research, and clinical evaluation. He has contributed to the development of treatment guidelines for snakebite in India and is passionate about improving antivenom efficacy and safety. Over to you, Dr. Kadilka.
Yes, thank you very much, Ali, and very good evening to fellow panelists. It is my pleasure to be here, and I'll be talking about snake antivenom, a poorly understood essential medicine. Now, when we talk about snake antivenom, it is one of the oldest biotechnological products being manufactured for over 100 years now. And it is a unique product in the sense that the raw materials required for manufacturing snake antivenom are both biologically derived. The first basic raw material required for snake antivenom manufacturing is snake venom, and another product is the hyperimmune plasma, which is produced in the horses. So you can say this is a very unique product where you use animals to produce a therapeutic product. So it is a form of artificial passive immunization used to protect life of a patient at a very short notice, which is generally seen in case of a medical emergency of snake bite. Antivenom is the preparation of either intact or fragmented immunoglobulin G. So there are some products which consist of whole immunoglobulin G, some are fractionated to yield FAB2 prime fraction or FAP fraction. And these are used for the treatment of severe envenoming from the bites of venomous animals. And these ready-made venom-specific antibodies are raised in animals, and mostly two host animals are used for producing hyperimmune plasma, either equines or sheep. The snake antivenom is a true lifesaver. If you administer snake antivenom as early as possible after the bite, it can definitely save the life of the person. But unfortunately, always in the news is the failure of antivenom, and that is why I want to emphasize why it is a poorly understood therapy. Antivenom is the only specific time-tested and proven therapy available for the treatment of snake venom. Now, if we look at the snake antivenom manufacturing process, it is a very lengthy and laborious process, and it starts with the horse procurement, because almost 95% of the commercial antivenom manufactured and available in the market are derived from horses. So the first step is horse procurement. After procuring the horses, they are kept in quarantine or isolation to ascertain their health condition. And once all the health parameters of the horses confirm to the acceptance level, then the horses are taken up for production. The production starts with the primary immunization phase, wherein increasing sublethal doses of snake venoms are injected at periodic intervals. And just to tell you, the doses in the beginning are in micrograms, which are subsequently increased slowly over a period of 4 to 6 months to a milligram level. So it is basically sensitizing the animals without causing any harm. After the initial phase of primary immunization, we test the blood plasma to ascertain whether particular level of antibodies have been generated by the minimum. We start collecting blood at regular interval, and in between we also give regular periodic boosters. So that cycle continues. Every month there will be some boosters to increase the titers and regular blood collection. After the blood is collected, the cellular component is of no use in the manufacturing of antivenom. So it is returned back to the same animal, and the plasma, which contains specific venom-specific antibodies, they are fractionated using enzymes and a chemical separation process. They are further concentrated to form the active pharmaceutical ingredient. That particular active pharmaceutical ingredient is formulated using stabilizers, preservative, and other salts. Then it is filled into the vials, and in case of lyophilization product, it is subjected to a process of lyophilization to convert it into a freeze-dried product. The advantage of lyophilization is that it increases the shelf life of the product, because typically a liquid antivenom product has a shelf life of 2 years, whereas the lyophilized product has got a shelf life of 4 years. And another most important advantage is that lyophilized product doesn't require cold chain. So that product can be stored or transported at ambient temperature, preferably below 30 degrees centigrade. After the product is manufactured, it is subjected to extensive quality checks, and then it is taken up for distribution. So if you look at this entire process, it takes about 6 to 7 months to get a product from animals after induction into the program. So this is some peculiarity of manufacturing of antivenom, and because of this, you can see that production of antivenom has to be planned well in advance. Even if there is increased demand, it is not possible to increase your production immediately, because that entire process takes a very long time. That's why many times you hear news that the antivenom is in shortage, because if in case of say flooding or in other emergency situation, if there is increased demand, the productions cannot be scaled up immediately. Now, what are the merits of antivenom? Prompt administration of anti-snake venom is of paramount importance for neutralization of unbound circulating snake venom components for early response to treatment. So it is very important. Administration of antivenom as early as possible after the bite ensures that all the circulating venom molecules are bound by the specific antibody, and that's how the further deleterious effects caused by the venom components can be avoided. And that is why snake antivenom is most effective when given intravenously, because they can directly bind to the venom molecule. And because of this reason, any delay in administration may result in increased dose requirement, because by the time the venom molecules get bound to various organs and they start causing some kind of damage, and in that case, to get response to treatment, more dose can be required, and that's how overall the effectiveness of the antivenom can reduce. Snake antivenom is effective to prevent or reverse most of the harmful effects of snake bite envenoming. When administered early, snake antivenom is not just life-saving, but can also spare patients from some of the suffering caused by necrotic and other toxins in snake venom, leading to faster recovery, obviously less time in hospital, and a more rapid transition back to productive life. So that's why snake antivenom is the only specific antidote for snake envenomation. But important point to be remembered here, that although snake antivenom is the only specific treatment, it is not the only treatment, because snake venom consists of many toxic fractions, and that's why it can affect multiple systems in the body. So a lot of supportive therapy is also equally important apart from administration of antivenom. Now, being a manufacturer of Panap Premium, I would like to introduce this product to you. This Panap Premium is recommended for the treatment of snake bites by African adders, carpet vipers, spitting cobras, neurotoxic cobras, and mambas. So practically all medically important snake species in sub-Saharan Africa can be treated by using Panap Premium. Now, Dr. Williams has just now explained the process of WHO risk-benefit assessment and listing. It is quite a comprehensive process, and any particular company and product goes through a thorough check at various levels, starting with comprehensive desk review of the dossier, independent laboratory evaluation of the product, GMP verification, audit of the manufacturing facility. So after going through all this, our manufacturing site was approved by WHO in November 2019. And after that, the product assessment took a very long time, because our product is a polyvalent product which is targeted against many snake species. So the assessment took lot of time, and ultimately the product Panap Premium was approved in March 2023. And it has been approved and recommended for the treatment of 24 medically important snake species in sub-Saharan Africa. Now, this product is specially developed in life form to avoid cold chain maintenance, because it is very difficult to maintain cold chain in rural areas, far-flung areas where electricity supply is always a big question. This product has a shelf life of 4 years. For the manufacturing, we are sourcing snake venoms from Africa, and this product is widely distributed in most of the sub-Saharan countries. And we have undertaken post-marketing surveillance of this particular product in many countries in Africa. Now, what is the dose recommendation? As explained by Dr. Williams, the dose of antivenom is calculated based on the number of vials required to neutralize average amount of venom that may be injected by a snake in a particular bite. So, after comprehensive preclinical studies in murine model and considering the average venom yield of that particular species, the doses recommended for Panap is that for carpet vipers, the dose is 1 to 3 vials. For African adders, it is 3 to 6 vials. Whereas for African spitting cobras, which inject very large amount of venom, the dose recommended is 20 to 40 vials. This is for non-neurotoxic envenomation syndrome. And for neurotoxic envenoming syndrome, if it is by mambas, the dose recommended is 10 to 25 vials, whereas for neurotoxic cobra, the recommended dose is 20 to 40 vials. Now, since it is a polyvalent product, Pan-African region, we try and source venom from many origins within Africa so as to add antigenic variation, because as you know, the snake venom has got both regional variation as well as seasonal variation. So to address that antigenic variability and so as to make product effective over a very large area, we try and procure venoms from many possible countries from all over Africa. So this is the list of countries from where we source different venoms for the manufacturing, and that's why ultimately it is showing good results almost all over the African continent. This product has been extensively studied by independent research groups, by preclinical studies, and almost everyone has given very favorable report about its preclinical effectiveness against different venoms. Now, this is the list of snake species for which Panap has been recommended. The top chart gives 24 snake species that was studied and recommended by WHO. So, on the left hand side is all Naja. They are all cobra, including both neurotoxic and spitting cobras. So it is recommended against 12 Naja species, then 4, that is all 4 mamba species, 3 adders, and 4 carpet vipers. Additionally, in another study, it was shown to be effective in cross-neutralization of the venoms of 4 more Naja species, that is Naja ashei, Naja nubai, Naja nivea, and Naja woodai. So overall, this product is effective against almost all medically important snake species that are prevalent in sub-Saharan Africa. Now, if we look at the status of Panap in Kenya, the product is registered with PPB in Kenya. It has been supplied to KEMSA and is widely distributed in healthcare facilities all over the country. We are on our own have also carried out some training of healthcare workers so as to make them aware about the use of the product in key locations. And currently a post-marketing surveillance study is going on in India. Our distributor in Kenya is Nairobi Enterprises, which is in Nairobi. If we look at the status of Panap Premium in East African countries, our product has been supplied in most of the countries in East Africa, including South Sudan, where WHO procured the product and it supplied to South Sudan. Now, Panap Premium has been widely distributed everywhere. You can see it is almost reach almost every country in sub-Saharan Africa, and it is also regularly supplied through UNICEF to many other countries, UNICEF, MSF, that is Doctors Beyond Borders, and WHO. Now I'll show you an interesting case report, like why that giving initial high dose is important. This was a case in South Africa a few months back. A very large black mamba, 2.4 meter in length, bit a person on thigh and it reached the healthcare facility within 42 minutes and started showing rapid onset of neurotoxic symptoms. So considering the positive history and likelihood of developing a serious consequence of that thing, the doctors over there took a decision and administered 15 vials of Panap Premium in 30 minutes. And in spite of bite by such a big snake, the patient could recover uneventfully and made a successful recovery. So this is a success story of one example I wanted to show you. So that's all from my side. Thanks for your attention and thanks Ali for providing me this opportunity. Thank you. Back to you.
Thank you very much, Dr. Kaldika. That was very interesting. And it's also good for us to have the contacts. If possible, what I will do is I will put the direct contacts of the premium distributors in the WhatsApp group so that people can contact them directly. We are being asked, is Premium Panaf available in Zambia?
Yes, it is available. I can send you the details of our distributors in Zambia.
Thank you very much. And in this country, for people that are saying that they're not able to get antivenom when they need it, what is what should they do about that? What can people do to try and make sure that antivenom is being stocked, particularly in the high-risk counties where there's problems?
Actually, we are working closely with KEMSA and Ministry of Health, try to find out what is the actual requirement. The main problem about snakebite is that we don't have exact epidemiological data. So we are reaching out to counties, trying to find out what is the requirement. And for any requirement, I have here, Mr. Sanjay is here, who is the representative of our distributor in Kenya. So you can give their contact number. If anybody is interested in procuring Panaf in Kenya, they can contact our distributors and it can be made available.
Okay, they can also contact if you go to any of the information that you've received. I'm going to type my email into the chat box right now. If you contact me, I will be able to send you the contacts for the Premium Panaf distributors. Please send me an email and I will respond straight away. The estimated cost per vial is about $60 per vial. Well, I was just asked whether there was antivenom in Mozambique. But all of these questions we can answer. The one question that has been asked a couple of times is if antivenom is expired. Thankfully, the premium product, as Dr. Kadilka just told us, it's a dry product. So thankfully, it has a shelf life of about 4 years and it does not need to be refrigerated, which is a very big plus point for us in Africa. So what do you say about in emergency circumstances, if antivenom is expired, what is your suggestion for that?
I can tell you it retains potency, it can work, but it has got its own legal issues. So technically speaking, it cannot be recommended.
I understand totally. All right, Dr. Kadilka, thank you very much indeed for joining us tonight. I would now, last but not least, like to welcome Dr. Mitchell Okumu to present a situation report on the post-marketing surveillance of Premium Panaf, which is currently happening here in Kenya, and we're all very excited to see how that's going. Dr. Mitchell Okumu is a certified forensic pharmacist and toxicologist registered with the Pharmacy and Poisons Board of Kenya with 10 years of snakebite research experience. He holds a PhD in pharmacology and toxicology from the University of Nairobi and is the secretary of the International Society of Toxinology's Africa and Middle East section. He's also a member of the Royal Society of Tropical Medicine and Hygiene, the Toxicology Society of Kenya, and the Pharmaceutical Society of Kenya. Dr. Mitchell Okumu, we are really interested to see how this surveillance is going, and we're looking forward to hearing from you.
Yeah, thank you very much, Ali. As you heard, my name is Mitchell Okumu. I'm a pharmacist, and I've been involved in snakebite research for some time now. Okay, so thank you very much for the opportunity to present some work on what we've been doing around post-marketing surveillance in Kenya. So the title of my presentation today will be a situational report on the post-marketing surveillance study of PANAF Premium. So I think it's important to just lay some foundation. Some of this was already presented earlier, but I think it's important to provide context on why it is absolutely important to have antivenom. So we have about 2.7 million envenomings every year, resulting in about 138,000 deaths. And as you can see from this map, you can see that Africa and the Middle East is among the most affected in terms of envenomings, where we get about 580,000 and about 32,000 deaths. And out of these, I think it's important that in the previous presenters have talked about mortality, but there are also other sequelae that result from snakebite. So in Africa, we have Nigeria leading in terms of the number of snakebite cases at 43,000. They also lead with regard to the number of deaths, the number of amputations, the number of post-traumatic stress disorders, and this is followed by Ethiopia, Congo, Kenya, Uganda, Tanzania, Sudan, and South Africa. So these are the top 7 countries where snakebite is an issue. And you can see Kenya, we are firmly in the middle with about 15,000. But again, these are rough estimates because, as you know, snakebite is often underreported. So the 3 major syndromes of envenoming, I saw one of the queries on the chat box was, what is the indication for antivenom? So this is it. We have 3 major syndromes, and according to the WHO, they say it's about 6, but the Medicines Sans Frontiers are the ones who came up with this table. So we have the 3 major syndromes as cytotoxic, hemotoxic, and neurotoxic. So the cytotoxic syndrome is characterized by painful progressive swelling and necrosis. And here some of the species that are responsible for this include the spitting cobras and the adders, as well as the saw-scaled or carpet vipers. Then as far as the hemotoxic syndrome is concerned, it's also known as the viperid syndrome. So here you'll have a lot of bleeding, non-clotting. This is caused by the saw-scale viper, the carpet vipers, and sometimes the Bitis species, and the boomslang. Then for the neurotoxic syndrome, also known as the elapid syndrome, this is characterized by progressive weakness and paralysis, and the species that are involved include the non-spitting cobras and the mambas. So I won't go over this slide because this has already been presented by the previous presenters. But I think it's important that we understand where we've come from as a continent. And the issue of antivenom is at the heart of snakebite management. And because snakebite is a neglected tropical disease, there's not much impetus for pharmaceutical companies to produce antivenom. So we've had quite a number of antivenoms over the years, and this is exemplified by this table. So between 2014 and 2018, these are some of the products that were available at that time. So we have the Asna antivenom, and these have been manufactured by different manufacturers around the world. And you can see from this table that we have only 2 products that are manufactured in Africa. So that is the South African Institute of Medical Research polyvalent antivenom and Vaxera, which is currently not available. So it's, as you've heard from the previous presenters, it's quite an expensive undertaking to manufacture antivenom. But I'm pleased to inform you that Kenya is on the cusp of having a facility to be able to manufacture antivenom. In the initial stages, it will be fill and finish, but subsequently, hopefully we can be able to manufacture antivenom in Kenya. So the major players in the sub-Saharan market has been Bharat Serum, Instituto Clodomiro Picado, Innocent Biopharma, Instituto Bioclone, owned by Sinales, then Premium Serums, Sanofi Pasteur, SAVP, and Vince Bioproducts. So the other players have been Micropharm, and basically they're developing the product Equitab, which is monospecific in nature. The other monospecific antivenoms include the South African Institute of Medical Research Boomslang antivenom, and the antivenom indicated for Echis ocellatus by Vince Bioproducts. So as you've heard from David Williams, antivenoms are not the same. So the difference comes as a result of 4 key factors. One is the immunizing mixture, what it is made up of, how the manufacturer adopts immunization protocols, how they fractionate and purify the product, and whether they fragment or concentrate the immunoglobulins. And I think this is where the problem of antivenom comes about, because the process is very expensive. Many manufacturers would want to cut corners because it's a very expensive undertaking to do all this, and it takes a lot of time. So as you heard from David Williams, WHO recommends that snake antivenom is evaluated both preclinically and clinically. But in all honesty, I think in terms of clinical evaluation, it becomes really difficult to have an absolute clinical trial, because unlike other products, it's very difficult to have or to recruit participants who are knowingly going to be envenomed and then get the antivenom. So that has been the biggest problem with regard to having randomized clinical trials. Notwithstanding, SHIPO in 2010 came up with a categorization of clinical studies, and these have various extents. They provide different quality of information. So we have the anecdotal clinical reports. So these are usually, most of the time, retrospective in nature, but they may also be prospective, basically looking at clinical effectiveness and safety. So you'll find the sample size will be small. Then we have the observational cohort study. It's also prospective in nature, also looking at therapeutic efficacy. Then we have the non-randomized comparative clinical studies. These also look at safety and efficacy, and they're prospective in nature. However, patient allocation is not random and depends on other factors. Then we have the randomized clinical trials, but these ones are really difficult to do because you can imagine it's not very easy for manufacturers to collaborate and have an RCT. That is one. Then number 2, getting funding to be able to do a randomized clinical trial is also a big problem, because snakebite is already underfunded. So unless it is a manufacturer who has 2 different products, as has been the case with Equitab Plus and Equitab G, then it becomes easy to do this kind of randomized clinical trial, where you can be able to randomize patient allocation, and you can basically be able to tell which product is better than the other. So in terms of quality, anecdotal clinical reports are at the lower spectrum of quality, Then we have the observational cohort study and the non-randomized comparative clinical studies to be of moderate quality. Then the highest quality, of course, is the randomized clinical trial. So bearing in mind this, we set out together with my colleague, Dr. Eugene Erulu from Watamu Hospital, and a few other healthcare practitioners at Jaramogi Oginga Odinga Teaching and Referral Hospital in Western Kenya, Marigat Sub-County Hospital in Baringo, and Malindi Sub-County Hospital in Kilifi to basically try and see whether the product that we have currently in the market is safe and is efficacious. So these 3 sites were selected based on persistent media reports and also literature that is available. As I had elaborated in the introductory slides, snakebite is underreported. And so we can only rely on what we are able to get, if that is gray literature or if it is the scientific literature. So these are the 3 sites that we selected, and our objectives are basically to document what are the clinical syndromes among patients who present to these 3 facilities, what are the early and late adverse events following the administration of the product, Panaf Premium, then also to determine how many vials of Panaf Premium is required to reverse some of the clinical symptomatology of among snakebite victims that present to the 3 hospitals. And then ultimately, we also wanted to see how long it took the patients to recover. So this is currently ongoing. We have managed to recruit 57 patients, and it's just according to standards of care, the guidelines for management of snakebite in Kenya. That's what we are using for this study. So basically, we started with a courtesy call at the Jaramogi Oginga Odinga Teaching and Referral Hospital. So the picture on the left is the team, part of the team from the hospital, meeting the chief executive officer just to brief him about the study and the objectives of the study. And then the picture on the right is the study team members. That is Dr. Opanga, Dr. Vincent, Mr. Geoffrey Kapesa, Ms. Cynthia, and Dr. Erulu, who is the principal investigator. So after that, we went to Baringo. So here again, we had a site activation visit, and you can see the picture on the extreme left is Dr. Eugene Erulu, the principal investigator, taking the healthcare practitioners. These are nurses, clinical officers, medical officers through snakebite management protocols. And then the picture in the middle is the county executive, so the Minister of Health at the county level and his team, that is the Director of Public Health and Director of Medical Services, and our county coordinator, Dr. Robert Rono, who presented earlier. Then finally, we had a site activation visit as well in Kilifi, where we met the county leadership, including the county pharmacist, Dr. Cecilia Nanjala, the county public health officer, and our county coordinator, Dr. Mangongo, who is coordinating activities at the county. Then we also had a training session with the healthcare workers as well, just to take them through snakebite management protocols. So once we did that, so this is just a photo of the product, Panaf Premium. So it is a 20 ml vial, 10 ml diluent. And this is now the approval from PPB, Drug Retention License for the product. And these are the approvals, as you heard from David Williams. You start with the WHO pre-qualification, then you have the registration and approval by the Pharmacy and Poisons Board. Then we went through the ethics that the Jaramogi Oginga Odinga Teaching and Referral Hospital Institutional Scientific and Ethics Review Committee. Then we also got approvals from the National Commission for Science, Technology and Innovation. And as I have shown on the slides, we were able to meet the county leadership and got approvals to also to begin the study in those 3 counties, that is Kisumu County, Baringo, and Kilifi. So basically, this is what we do in terms of management. So just observe the patient when the patient presents, whether they have any cardinal signs and symptoms of envenomation. And as I've shown already in the previous slides, the syndromes, that is cytotoxic, hemotoxic, and neurotoxic. And we do the 20-minute whole blood clotting test for all the patients that present. This is basically to check for coagulability of the blood. Then I've seen David Williams' presentation on using 200 mils. Dr. Timothy Hardcastle also talked about that in the last session. But according to our guidelines, we have been using 500 ml normal saline, diluting the antivenom in 500 ml of normal saline. Then again, the administration has been through slow IV infusion over 30 minutes to an hour at 15 to 30 drops per minute. And this has been increased gradually if there's no reaction. Then we've been monitoring vital signs every 15 minutes for the first hour. And we've been very keen on whether there's been any development of a rash, any restlessness from the patient, any wheezing, shock, hypotension, or any other allergic reaction. For instances where we've observed ADRs, infusion was stopped briefly, vital signs were monitored, and we managed the patient using supportive care, oxygen support, and adrenaline. Then once that has been managed, antivenom has been restarted slowly and gradually, and the rate has been increased with time. Then vital signs continue to be monitored over 4 to 6 hours. And we've also offered supportive care, including tetanus toxoid, paracetamol, and then wound care for the patients. Then we developed a tool where we collect various variables. And because paper-based data collection is very problematic, you can often run into missing data. We onboarded that onto a Kobo Collect toolbox, and each of the sites has a designated data collector and someone who deputizes them. So every entry is filled on the physical forms, and then that is onboarded onto the Kobo Collect tool. And we can be able to monitor real time and troubleshoot in case the data has issues. So in terms of results, as I said, we have about 57 cases. So 6 cases are yet to be onboarded, but this is basically how the results look like. So we have 26 cases in Baringo, we have 13 cases in Kisumu, and we have 12 cases in Malindi. This is the age distribution. So in Baringo, we are seeing most people are between 30 to 39. In Kisumu, 20 to 29. And in Malindi, we are seeing most of the patients are between 10 and 19. Then in terms of the gender, there's skewed towards the males. So we have more males in Baringo, Kisumu, and Malindi being bitten. Then in terms of the type of snake, regrettably, we have not been able to identify 16 cases of the bites of the snakes that bit the patients in Baringo, 12 in Kisumu and Malindi respectively. And this underscores the fact that it's absolutely important to use the syndromes in the management of snake bite rather than the identity of the snake, because not everyone is able to identify the snakes. But through the Biocan Snake Farm, we have been able to get support from Kyle to be able to identify some of the snakes. So we've had about 5 or 6 incidences where some of the snakebite victims or their families have killed the snakes and have come with the snakes to the hospital. And that's why it was easy to identify some of them. We also observed that we had a lot of red spitting cobra bites, particularly in Baringo. And we had one case of a black mamba bite in Kisumu. Then in terms of the clinical syndromes, this is what we observed at JOTRH. We saw cytotoxic, neurotoxic, and hemotoxic syndromes. And in Marigat, we saw both cytotoxic and neurotoxic. Then in Malindi, it was primarily cytotoxic. Then in terms of adverse drug reactions, so these are categorized into 2. So we have the early adverse events, which come anywhere between 3 minutes to an hour after the administration of the antivenom. And then we have the late adverse reaction. So we are actually following up on the patients up to 30 days. So the data collectors and their deputies usually call the patients once they're discharged, and we are able to track through follow-up whether they've had any adverse events. So at JOTRH, we have observed early adverse events, including itching, tachycardia, fever, urticaria, chills, hypotension, and sweating. And these we have been able to manage relatively easily. Then at Marigat, we have observed urticaria, fever, itching, hypotension, and tachycardia. And this was particularly in 3 patients. Then Malindi, we got itching and a patient complained of backache. Then in terms of the number of vials used, so we found that Baringo, and this is corroborated by the persistent media reports, and people in Kenya appreciate that Baringo is one place that there's a lot of snake bites, and that is illustrated in this table, where so far 115 vials have been used in the 26 patients who have been treated there. Then we've had 24 vials being used at JOTRH, then 36 at Malindi. And the mean number of vials at JOTRH is about 2. At Marigat is about 4, and at Malindi is about 3. Then in terms of comparing the 3 sites, the number of vials across the 3 sites. So here we did one-way ANOVA and used Tukey's test as a post hoc, basically to compare the 3 sites and see where the consumption how the consumption is relative to each other. So here you can see when you compare Marigat and JOTRH, you can see that JOTRH uses significantly lower number of vials as opposed to Marigat. The same is true for the comparison between Marigat and Malindi. And as you can see, there's no significant difference between the number of vials used at JOTRH and the number of vials used in Malindi. Then in terms of the length of hospital stay, we were able to see that at least 2 days, that was the number of days that people had at the hospital. Then in Marigat, we had about 4, and in Malindi was 8. But this data is a bit skewed because there was a patient who was in the hospital in Malindi for 38 days. And ultimately, they had to be amputated. And that is why the patient stayed for a long time. So this result, you can take it with a pinch of salt, because this has to be corroborated with the number of vials. Then this is also a comparison in the length of stay. So this is a one-way ANOVA and Tukey's post hoc comparison. So between Marigat and JOTRH, no difference. Then between Marigat and Malindi, there's a difference. And then between JOTRH and Malindi, there's a difference. So we are seeing that Malindi, because of that patient that stayed for a relatively long period of time, you can see that the the number of days or the length of stay is quite high. These are the references, and thank you very much for your attention. And thank you, Ali, for organizing this, and back to you.
Thank you very much, Mitchell. That was really interesting. There's some questions. Somebody asked why, I mean, it's not a question I think you can answer, but, you know, why when in our region down here at the coast, we have all of the varieties of snakes, but it seemed like only the cytotoxic snakes were biting at the time. I'm not sure you can really answer that question. I think it's just a situational thing.
Yeah, I think I can give it a try. You know, snake bite is usually seasonal, and we started this trial, as you saw in the slides. We started it about in November. So we've been having, like, now is a rainy season in some parts of the country, and we expect the number of bites to be higher, and we expect the diversity of the bites also to be a bit, you know, more. Because when we started, it was very slow. We got a few cases here and there, one case here, two cases there. And there's a very interesting dynamic, because when you go to the drier places like Kitui, Baringo, you find there that when it is dry, they have more bites. But when you go to Kisumu, Kilifi, Mombasa, you get more bites during the rainy season. So there are those interesting dynamics that are there. We've not fully explored, you know, all the number of bites that we could, but the study is still ongoing. So probably in the fullness of time, we can get more diversity.
Okay. Thank you very much. Can I just invite Dr. Williams back to the floor? He had a couple of things he wanted to add to his presentation.
Yeah, hi, Ali. Mitchell talked about the issue of clinical trials, and one of the things that WHO has been exploring as an alternative to expensive clinical trials is what's called a monitored emergency use authorization of snake antivenoms. It's likely to become part of the next lot of antivenom guidelines that we publish in 2026. It's based around the same proven ethical framework developed for Ebola in 2014 in West Africa, and has been used previously with clinical studies of new treatments for COVID. It's a fairly simple approach. There are some requirements which I can sort of just jump to very quickly. So the criteria that no proven effective treatment exists in the setting, and certainly, you know, evidence of effectiveness in many antivenoms is often absent. It's not possible to initiate clinical studies immediately, and again, that's something that can be demonstrated. Clinical trials are exorbitantly expensive, difficult to manage both logistically and administratively and ethically, to the point where they're probably simply unaffordable. There must be data that gives some preliminary support of the intervention's efficacy, such that it can be put to a scientific advisory committee. And again, the WHO risk-benefit assessments can provide that evidence. And then the country authorities have to have an appropriately qualified ethics committee. And again, most countries have ethics committees that would be able to deal with this. There need to be resources to minimise risks as much as possible. And again, this is something that a collaborative effort on the part of manufacturers, researchers, ministries of health, regulators and other partners could ensure. There must be informed consent. So it's not that much different to a clinical trial in that perspective. And again, the emergency use of the intervention is monitored, the results are documented, shared in a timely manner with the wider medical and scientific community. And basically it means that you could take a product, you can go through the process of setting this up, and then make it available in the marketplace and very closely monitor its use, determine the dose that's safe and effective, and make modifications in real time to how you use the product till you reach the optimum. It would give you the ability to much more rapidly collect data and make some meaningful recommendations at substantially lower cost. And of course, making sure that you actually have evidence from all of the people who need it. In a clinical trial, often children, pregnant women, the elderly will be excluded for various reasons. They're actually some of the people who are most likely to need antivenom that really works. And so they really should be included in the way we study antivenom use. In fact, ICH, the International Commission on Harmonisation, has a new protocol that's under public consultation at the moment on recruitment of pregnant women and lactating mothers to clinical trials, which does put forward a very strong ethical argument for their involvement. So that was one thing I wanted to mention, is that there is an alternative to being able to collect evidence in a cost-effective manner. But the other one, which I thought people might want to hear a little bit about, is that WHO is working at the moment to list the products that we have assessed in our procurement system, and to develop a pooled procurement project where countries would be able to source antivenoms through WHO's procurement hubs, substantially lower costs than what they do at the moment, substantially larger volumes by being able to collectively purchase large batches of antivenom at a single time. It gives manufacturers some benefits in that they have only one person who's going to cut them a check at the end of the day to pay for it. WHO would pick up the product from their door and deliver it to the countries that need it in the amounts that those countries have ordered within 60 days. And the countries would eventually reimburse WHO for what they've used. They'd also provide an additional emergency facility for urgent resupply in disasters such as flooding events and other natural disasters. It would go hand in hand with a number of different things to help stabilise the market, including training of health workers, improved epidemiological surveillance, improved community engagement activities, which would basically be led by local people in the countries themselves, with some technical support from WHO and from the Ministries of Health. So this is likely to begin sometime later this year. WHO is currently going through the process of negotiating long-term supply agreements with the approved manufacturers. Products will then go into the WHO catalogue. 4 times a year, countries will be able to place an order that again will be shipped to them within 60 days. So 4 times a year, they will know that the product is actually going to turn up in the quantities that they've requested. So hopefully that's a little bit of an additional positive to carry forward with, for people who are listening in.
Thank you. Sorry. That's great to end on such a positive note. Thank you very much, Dr. Williams. I've just put my email address into the chat group. If your question was not answered, it's probably because I wasn't able to answer it or someone else didn't get to it. If you feel like you still want your questions answered, please email them to me. Follow that link, that email address, sorry. If you go back through the chat group, you'll find the link to join up the WhatsApp group. Thank you ever so much, everybody. It was a fascinating evening. I hope you've all learned a ton and enjoyed it as much as I did. Look forward to seeing you next Thursday, same time. We will be welcoming Professor Sir David Worrell and Dr. Eugene Irulu to talk to us about the clinical management of snakebite. And I'm sure it will be another excellent evening. Have a great evening. See you all next Thursday.